Okay, sir.
Yeah, good evening. I hope I'm audible. So, in this week's problem solving sessions, we'll be looking at a few sample problems related to week 5's lecture tutorials. So, to start with, the first question is which of the following agents is used to make E. coli competent cells? And four options are given. Option A, mitomycin C. Option B, UV light. Option C, calcium chloride. Option D, ciprofloxacin. Like we usually do, you can take a couple of minutes and then try to start eliminating each option. And if you know the right answer, we can also discuss the mechanism with which that particular agent helps in making E. coli cells competent. So whoever wants to answer can raise your hand after a couple of minutes. So since only three of you are here, what we can do is you one of you can start by explaining what competent cells are. We'll take it from there. Rajata, do you want to go first? Okay, Chandrima. Yeah, Chandrima. What are competent cells?
So competent cells are those cells which are able to take up the DNA, the recombinant DNA, taking that.
Should it only be recombinant DNA?
It can be any actually. It's not only recombinant, any foreign DNA exactly.
Right.
That
It
is.
can be any DNA, any external DNA, anything from its environment. Right. Okay. So with respect to that, what modification do you think cells should undergo so that they can take up recombinant DNA or any foreign DNA?
So they have to work on their surface chemistry means the charge present on the cell membrane,
Hmm.
so that they could neutralize it, so that the DNA could enter there
Okay.
from there.
So you are talking with respect to prokaryotes, specifically E. coli, right?
Yes.
Okay.
Yeah.
Okay. Yeah, right. Like you had mentioned, competent cells are those cells that are capable of taking up any external DNA. And in order to make any kind of cell competent, we need to work around the cell wall and the cell membrane. So that would depend on what are the components at the uh, external periphery of the cell. With respect to E. coli, for a particular method, we are working on the chemistry. So, because you mentioned about the charge, what is the charge of the cell membrane in E. coli? Others can also try to answer if you know this, Vishwajit and Rajata, if you know what is the charge in the cell membrane.
Negatively charged
Okay, why do you think it's negatively charged?
due to the presence of phospholipids, lipopolysaccharides.
Hmm. So, lipopolysaccharides make it negatively charged. Is it? Yeah. Did someone raise your hand or got a notification? Yeah. Yeah, Vishwajit.
Sir, uh, uh, it, membrane has a positive charge
Why do you think it has positively charged? Why do you think it's positively charged?
uh, because um because DNA has a negative charge, so it attracts. That's
But is the you can think about it this way. What do you think is the function of cell membrane?
function of the cell membrane is to protect cell from any I mean foreign particle or yeah.
Huh. So if that's the case, why would it want to attract DNA?
Sir, polar group can attract. I mean, the uh, hydrophilic molecule can attract DNA.
Yeah, but so you need to think about these with respect to the function of it. Like you told that cell membrane is like guarding the cell from external substances. It could be anything, like from osmotic pressure to other stuff. So if that's the case, should it really focus on attracting DNA? Vishwasthi, should it
No, sir.
it shouldn't. Okay. Yeah. So like so the cell membrane is negatively charged. Okay, that is clear. Now with respect to the reason for it, like you have different phosphor groups, phosphatidyl, serine, phosphatinositol, and those colon groups. So these things could impart a negative charge. So Rajata, you were right about that. Yeah. Now we'll come to the options. So we can start eliminating the options. Does anyone want to go first? Rajata? Yeah. Rajata first and then Chandrima.
Okay, so option D, I want to eliminate option B
Option
because
D.
it's an yes, sir, because it's
Okay.
an antibiotic that inhibits bacterial DNA gyrase.
Right. So ciprofloxacin inhibits DNA gyrase. Okay. Fine. Do you know what DNA gyrase does?
Not really, sir.
Okay. So that's a protein that will help in removing the supercoiling or basically reduce or improve the helicity such that there is very little torsion force in the DNA during replication. So you can check about that.
I guess it's there as a little amount in type 2 topoisomerase.
Yeah, topoisomerases also have something to do with the coiling topology of the DNA. Gyrase also has a similar role, but I think it will introduce negative supercoils that will relieve the stress that is caused because of torsion. So you can assume the DNA like a strand. Now during replication, what will happen is you have this replication machinery that will twist and turn the DNA. So when you have two strands that are bound together, if you twist one strand, that will create some stress on some other region. Now this DNA gyrase will help in relieving that. So that is the function of DNA gyrase. And ciprofloxacin prevents DNA gyrase from functioning. So it will affect replication. So that's how it is related. Ha
Okay, sir.
Chandrama. Ha Chandrama. So what option would you want to talk about?
Option A, mitomycin C. It is also an antibiotic.
Okay, mitomycin C. Yeah. And Vishwajit?
Sir, option B, UV light.
UV light. Okay, UV light would also not help them. So what? So do you guys say calcium chloride helps? Does anyone know how calcium chloride is helping for the E. coli to make E. coli cells competent?
It helps in neutralizing the negatively charged DNA and cell membrane.
Okay. So if it neutralizes the negatively charged DNA and the cell membrane, what will happen? So what do you think will happen when the charges are neutralized? Okay, I have an uh, image that would explain this better. So you have your cell membrane. Okay, I'll briefly explain the process of competent cell making. So in your vial, which contains this E. coli cells, you will have a trace amount of magnesium chloride, some calcium chloride, some glycerol, and your cells. So glycerol here will act as the cryoprotectant. You store your cells at a very negative temperature, like -80 or -60. You need magnesium chloride because magnesium, in a way, is a cofactor for your DNA polymerase and many other proteins which the cell needs. So you would add it just to ensure that DNA replication is not inhibited. With respect to calcium chloride, like you had mentioned, it would ensure your the negatively charged membrane and the negatively charged DNA are not repelled. Now your DNA is negatively charged, your membrane is negatively charged. Now, because of calcium chloride, which acts like an intermediate bridge, it would ensure that the repulsion doesn't happen, and you can in a way think of it like one calcium will be able to bind to both the DNA and the cell membrane. Calcium has a valency of 2, so it can make such bonds such that it physically interacts with both your membrane and the DNA. And when you give a heat shock, which is at 42 degrees Celsius, what will happen is the pores on the cell membrane will occur. Now, the already equilibrated DNA calcium and your cell membrane will be in such a position such that the DNA can enter the cell. After you give a heat shock at 42 degrees Celsius, you will keep it on ice for a few minutes. At that time, these pores will close. So this way, you will ensure that the DNA is entering the cell. So this is the mechanism by which DNA enters the cell. Was it clear?
Yes, sir.
Okay, cool. The next question is, while making competent cells, it is essential that the bacteria is in option A lag phase, option B log phase, option C stationary phase, and option D logarithmic decline phase. Yeah, we can take a couple of minutes and then start answering this question. Yeah, does anyone want to answer this question?
Sir,
Yeah,
uh, sir, right option or wrong?
you can start with the right option itself. It's a straightforward question.
Option B.
Okay, why do you think it's the log phase?
Because in that phase, cell started divided in very rapidly,
Right.
and those are I mean uh, uh, a good say. I mean their efficiency is good. I mean
Hmm. So, okay, I'll rephrase it and I'll tell you how it works in lab. I'll give you the practical idea as to why it is essential. So, like you mentioned, log phase is the right answer. So, in log phase, you have cells in a particular optical density such that their actual doubling time is 20 minutes, meaning every 20 minutes you will have one generation of cell. From one cell, you'll get two cells, and so on. Now, how is it relevant to competent cells? Does anyone have an idea about it? Why competent cells should be in log phase? Is there any growth step involved further in after competent cells take up DNA? Does anyone know about it?
Sir, I didn't get the question. Could
Okay,
you repeat it
so
again?
yeah, yeah. So Rajendra, do you agree that log phase is the right phase in which competent cells should be made?
Yes, sir.
Okay, so that means it has something to do with the growth of the cell, right?
Yes, sir.
So, do you think where do you think this growth part comes in with respect to competency of the cells?
Sir, because here the replication stage, it is just doubling that DNA that time. So somehow it is linking that with the competency in this log phase. Actually, it is happening in the, so somehow it is there a link that the DNA repairing or DNA damage can be done in this phase. That's why it is essential for the bacteria to be in log phase.
Yeah, right. And adding on to
And also,
your,
since we are,
uh, yeah, Rajata, sorry.
sorry, sir. Also, adding on to that, since they are dividing,
Huh.
so their cell walls and cell membranes are more permeable at that time.
Mm, so how you have in the COMCEL is you don't have particularly any media. So, and they are frozen in glycerol. So we don't really know if it's permeable. I don't know. I can't comment on that. I can tell you what I know about this, but we can check that. If you have any references, you can maybe share that in the Google Meet chat. We can take a look at it at the end of the class once we're done with
No,
all the
sir, I just
questions.
told like that only. I'm also not sure. I just so it may be one of the reason for this.
Okay. Okay, but we can go with the idea that Chandrima was mentioning. So, how you can understand this? I was earlier telling you, right, that once it uh, once we add the DNA to the cell, we'll give it a heat shock, where the membrane will have some pores, the DNA will enter, then we'll keep it on ice, where the membrane will close. Is it clear till this point?
Yes, sir.
Yeah, so
Yes,
now
sir.
what we'll do is we'll add some media to the cell and we'll grow it for some time, like 1 hour. So 1 hour means 3 generations with respect to E. coli, right? So after this 3 generation, we expect that multiple cells have taken up the plasmid and they have replicated. So multiple generations of cells have this plasmid now. Now, how do we know that these cells have taken up the plasmid? So for that, what we'll do is we will take an agar plate with an antibiotic that's there in the plasmid. And we will plate these cells on that agar plate. And then only we will know, depending on the colonies, whether the bacteria were able to take up the DNA or not. So, in order for the selection process to be good enough, we want more number of cells to take up the DNA and more number of cells to retain the DNA. If we take it in the log phase, like it was mentioned by Chandrima, it's the DNA replication would happen at the right pace, and the cellular replication will also happen at the right pace of 20 minutes. So, in order to yield to get many colonies at the end of your selection, we would prefer it to be in the log phase. So that's the practical aspect of it. So was it clear? Yeah, you can let me know. If there are any questions, you can let me know. Otherwise, we'll go to the next question. Okay. Okay, then we'll go to the next question. Yeah, so this is a numerical question. So in an E. coli transformation experiment, 0.001 microgram of DNA was used to transform 100 microliters of competent cells. The cells were recovered by adding 900 microliters of LB media, resulting in a final volume of 1000 microliters. From the 1 ml of total reaction volume, 50 microliter was plated and subsequently 500 colonies were obtained. The transformation efficiency for the batch of competent cells is so yeah, if any one of you have done the calculation, you can share the method or we'll discuss this again. Does anyone want to go try this question? Has anyone tried this? Rajita, Chandrima, or Vishwajit? Uh, yeah, hi. So we are looking at the third question. So this question is on calculating the number of calculating the transformation efficiency. So here in this experiment, 1 nanogram of DNA was used to transform in 100 microliter of competent cells, and they got 500 colonies at the end of it. Now you want to find the transformation efficiency. Lichi, do you know how to approach this problem? Yeah, does anyone want to try? Chandrima?
Sir, actually, I'm calculating that.
Okay, so you know how to do this, is it?
Yes, sir.
Okay. What about Rajata and Vishwajit? Are you aware of how to do this?
Yes, sir. I'm
Yeah.
trying.
Okay. I'll just briefly go through the methodology. So to get the transformation efficiency, which is the number of colony forming units per microgram, what one has to know is the number of transformants, which is nothing but the number of colonies. In this case, it is 500 and the microgram of DNA. So they used 0.001 micrograms of DNA. Then what is the final volume at recovery? That is 1 ml versus the volume that is plated, that is 50 microliters. So here it's 1 ml by 50 microliters, which is 20, and here it is 500 by 0.001 microgram. So 500 into 20 by 0.001 microgram. 500 into 20 is 10,000, which is 10 power 4 into 10 power 4 divided by 10 power minus 3 microgram. So that is 10 past 7. So if anyone gets a different answer, you can let me know. We can go through the methodology again.
So the answer is coming 10 to the power 7 microgram per that colony units
It's a,
CFU.
it's actually colony forming units per microgram. Just a units. Yeah. So you got it right. That's the right value you're supposed to get. What about Rajata and Vishwajit? You were able to get the same value?
Yes, sir.
Oh,
Option D.
okay. Cool.
Sir,
Yeah, Vishwajit.
can you explain it once again?
Yeah, sure. So the formula that you need to use is number of transformants per microgram into final volume at recovery by the volume plated. So the number of transformants that you have here is 500 transformants, and the amount of DNA that you took is 0.001 microgram. Multiply that with final volume at recovery, that is 1 ml, divided by the volume that is plated, that is 50 microliters. So if you write this down, you will have here 500 divided by 0.001 into 1 ml divided by 50 microliters. Now 1 ml divided by 50 microliters is 20. And here you have 500 divided by 0.001. So that is 500 into 10 power 3 into 20. So that will give you 10 power 7. Should I make like write this down on the slide?
Okay. Got it, sir.
You got it?
Sir, actually, I wrote microgram DNA upon number of transformants.
Okay, you inverted it. Okay. Cool. We'll go to the next question. The next question is transformation efficiency in E. coli is highest for option A supercoiled DNA, option B closed circular DNA, option C open circular DNA, and option D linear DNA. So this is based on the topology of the DNA. Yeah, Vishwajit.
Sir, option A.
Hmm. Why do you think it's option A? Yeah, Vishwajit, you can unmute and talk. Why do you think it's option A?
Uh, Sir, I just remember that in lecture, sir, in the video,
Hmm.
the supercoiled DNA has highest efficiency for transformation.
Okay, but do you think can you think about the DNA's topology and what we discussed about the mechanism for calcium chloride method?
No, sir.
You can try thinking about it. Meanwhile, we'll talk to Chandrima. We'll get back to you. Okay? Yeah, Chandrima.
Actually, sir, if the in the case of supercoiled DNA, it is highly coiled, which makes the makes the DNA very compact, and
Hmm.
it helps to enter that DNA into the cell easily than other ones, because it makes, if it is compact, it will be more easier to get inserted in the E. coli. That is why supercoiled DNA transformation efficiency is highest.
Right. So, do you also think there is an order to this? Like the, okay, if that's the case, what you meant is right. So, can you now try to order the four options here based on highest transformation efficiency to lowest transformation efficiency?
Yes, first is similarly supercoiled,
Huh.
then the closed circular,
Huh.
then the open circular, then linear DNA. Similar, just like that option.
Okay. Now, adding on to this, do you think linear DNA will get transformed? Cells that take up linear DNA, do you think they will be transformed?
It is not that much easy for the transformation.
No,
That long chain of
let's say
nucleotides.
the DNA enters the cell and the pore is closed. Now, how easy it is for the cell to retain the linear DNA? We'll, for a moment, we'll forget about the physical difficulties that a linear DNA will have to enter the cell. We'll assume that this DNA has entered the cell. Now, how easy it is for the cell to maintain this DNA? Because that also is a part of the transformation efficiency, right? So, do you have any thoughts about it?
No, sir.
Think about it in terms of replication of the DNA. So, when you have a closed DNA compared to an open DNA, what is the difference that the DNA replication machinery will have when it is trying to replicate this? Yeah, Vishwajit, you had unmuted. Do you want to ask something?
No, sir. Sir, so linear DNA, uh, um, it cannot. In E. coli, no?
Hmm.
So it can directly, get transformed in E. coli. It is
I did not
easy.
understand. You're saying linear DNA, it's easy to transform, is it?
Yes, sir.
Why would you say that?
Because it is. It is in linear, no. So and other are in. Uh, I mean, super coiled and other things. So it needs. It should release and then it will bind to uh, um, the DNA of E. coli.
Okay, so think about it when you run the gel. You can, when you run an agarose, this is how it looks like for the different conformations of DNA. Your nicked DNA runs here, your linear DNA runs here, your supercoiled DNA runs here, and your single stranded circular DNA runs here. That would mean that the linear DNA is not compact enough, firstly. So, with respect to taking up of DNA, you need to think about how compact it is. Uh, Who was it? Chandrima. 1 minute, Chandrima. Yeah. So you need to think about how compact it is. By compact, what we try to say is, in a very small volume, the entire DNA should be folded and it should be present. If it is extended and if it has like projections, then it would be very difficult for the entire DNA to enter before the pore closes. Whereas if it is very small in with respect to the volume, then it can enter easily. That's how you need to think about this particular phenomenon. Was
Okay, sir.
huh,
Okay.
yeah, yeah, Chandrima.
Yeah, sir. In this case, actually, the linear DNA, it will be a more ori means origin of replication. But in case of a circular one, there is generally a single origin
No,
of
no. So
replication.
okay. So by linear, what we mean to say is you have the same plasmid, right? You just cut it at one point. So using an endonuclease, you cut it such that instead of it being circular, it is now open. That's the only thing. So the number of origins of replication is also maintained. There is no differences at the genomic sequence level. Is can you understand this, Chandrima? That's what we call as the linear DNA here in this
Yes, sir.
context. So in order to think about this, just try to think about how DNA replication happens. So when DNA replication happens, your one end is physically connected to the other end. You don't have linear DNA that survives within any cell. If you have linear DNA, it's very difficult for the DNA replication machinery to keep track of the ends and join it. Whereas circular DNA, your RNA polymerase, your DNA polymerase can just sit on the origin of replication, start replication, and end at the corresponding region. That doesn't happen with your linear DNA, because of which linear DNA cannot be replicated with ease. That is why it will be difficult for linear DNA to be, how do I say, to be replicated and maintained within the cell. So, independent of the compactness, what we have here, the order in which you mentioned was right, but the logic here is this way. Linear DNA cannot replicate, because of which the transformation efficiency will be close to zero. Was that clear, Chandrima?
Yes, sir.
Okay. Yeah, we'll then go to the next question. Which of the following components block the non-specific sites on the cell wall and increase efficiency of plasmid uptake in yeast cells?
Sir, can you just zoom in?
Yeah, sorry. So the question is, which of the following components block non-specific sites on the cell wall and increase efficiency of plasmid uptake in yeast cells? And we have four options: lithium acetate, single-stranded DNA, PEG 3500, and sodium fluoride. We can take a couple of minutes and then try to answer this question. Uh, Priya, so we are discussing this question. Which of the following components block the non-specific sites on the cell wall and increase efficiency of plasmid uptake in yeast cells? And the options are lithium acetate, single-stranded DNA, PEG 3500, and sodium fluoride. You can unmute if you know the answer, Priya.
Sir,
Yeah, sure.
can I?
Yeah,
Okay.
yeah, sure, sure, sure.
So, so option B is the wrong option.
Option B, single-stranded DNA,
Uh,
is it?
yes, sir.
Okay, why do you think so?
Uh, because it cannot, I mean, it is a single-stranded DNA, no? So it cannot block I mean, cell wall.
It's saying non-specific sites, so that means you don't have any specific region.
Huh.
So single-stranded DNA also has this phosphate backbone, right?
Yes.
So let's think about the logic that we earlier thought about, where uh, where you have these divalent ions that would ensure the negative charge on the DNA backbone and the cell membrane is neutralized. If that's the case, then maybe single-stranded DNA can help in. Don't you think so?
Maybe, sir.
Maybe. Okay, we
Uh,
have a set of possible
uh,
mechanisms, but we can, yeah, sorry. You were about to ask something.
Nothing. Just I'm. I mean, on right option, I'm confused between option A and option C.
Okay, what is option A, lithium acetate? What does it do? Yeah, Vishwajit.
Sir, I don't know, sir.
Okay, you can think about lithium in the same way like one thinks about calcium or magnesium, except that lithium is monovalent. You know that it is plus one
Yeah.
charge, right? So lithium will also neutralize your negative charge. That's what it does.
But acetate, it has um a CO.
So acetate's formula is CH3COO minus, right?
Huh.
Lithium is positively charged. So if you think about the interactions here, these are ionic. And like how you within water, okay, one thing that you need to understand about lithium acetate when it is in solution is if it is getting dissolved, that means the lithium charge and the acetate charge they are getting separated. Okay, like how salt dissolves in water. You have sodium chloride, Na plus and Cl minus, they initially have a bond. But when they are in water, they can dissolve. And once they dissolve, your Na plus and Cl minus are separate. They are no longer Na plus and Cl minus. They are separate entities. Do you understand this? Do you agree with this?
Yes, sir.
Similar is the case with lithium acetate. You will have lithium plus and acetate minus. And if you put DNA, the DNA's phosphate backbones negatively charged, that will be neutralized by the lithium. So that's how lithium acetate will play its function.
Okay, sir.
And what do you know about PEG 3500? Do you know what it is?
It is a polymer.
It's a polymer. Okay. Other than that, with respect to biological functions, can you think of something?
No, sir.
Okay. You mentioned that it's a polymer. 1 minute. Rajata raised her hand. We'll
Yeah.
listen to her. Ha, Chandrima.
In the case of PEG polyethylene glycol 3500,
Hmm.
it is actually bind helps to bind that DNA to the cell surface.
But how does it do? Do you know, like, what is the possible interactions?
No, that it could be from the charge due to any charge separation or due to this factor.
Not really. I'll explain that in a minute. Ha, Rajata, you had unmuted. Do you want to say something with respect to these options?
So I want you to tell the correct answer, which was, I guess, option B.
Okay, we'll come to that. Do you know how PEG 3500 works?
So I know it does not directly block non-specific sites. It
Hmm.
aids in DNA uptake process.
Yeah, but how does it aid? Do you know that?
I guess from creating a viscous environment. I'm not sure about this. I just heard it somewhere.
Okay. Uh, hmm. Yeah, Priya, you can unmute.
So it facilitates the deposition of plasmid and carrier DNA on the cell wall for easier uptake.
Hmm. Okay, so in general, how you can understand about these PEG is that these are polymers, you know that, and they have different molecular weights. In this case, what they show here is PEG 3500. That means the molecular weight is 3500 Daltons. Now, these pegs, they do not make it viscous, Rajata. If it makes it viscous, it is difficult for your plasmid or any biomolecule to move across the solution. You want to ensure that the viscosity is very little. You already have glycerol in it, so already your solution is very viscous. You can't make it more viscous. But what you need to do is, you need to here how PEG acts is, it acts as a crowding agent. It ensures that more of your DNA comes close to more of your cells. That's how PEG works. It doesn't physically cause any interaction like how calcium did. So that is the logic by which PEG would help any DNA or any molecule come closer. Now, these are large molecules and they cause this crowding phenomenon, making uh, biomolecules come close by. That's the functionality of PEG. Was that clear? Rajata, Priya, Chandrima, and Vishwadit, was it clear?
So can you repeat again?
Yeah, so these PEG molecules, these are, they do not really make the solution viscous. If it makes the solution viscous, you need to understand that the DNA will have difficulty moving near the cells if it is viscous. So it can't be viscous. Rather, it should aid in making the DNA freely move. And with respect to the functionality, these PEG molecules act as crowding agents, meaning many PEG molecules come close by and they ensure that DNA is concentrated near cells and vice versa. Cells are concentrated near DNA. So they basically increase the physical proximity between DNA and the cells by acting as a crowding reagent. Was that clear?
Yes, sir.
Okay,
Yes, sir.
okay. So now, yeah, Rajata, we'll come to the right answer. You were saying single-stranded DNA is the right answer, right?
Yes, sir.
Yeah. So now, yeah. So how do you think it works as the, how do you think it helps in blocking the non-specific sites, and how do you think it increases the efficiency?
It binds to non-specific sites on the cell wall,
Right.
hence preventing plasmid DNA or any DNA from sticking to those sites.
Okay, that is one. Huh? Do you also know of other possible phenomenon that can happen? What you mentioned is one of the possible mechanisms. Like if there are some regions that can interact with your plasmid DNA and just stick to the DNA but not allow it to enter the cell, the single stranded DNA will go occupy those sites in a non-specific manner. So your plasmid DNA will not bind to those regions but would rather go through the pores. And another method is there are nucleases, that is DNA cutting enzymes. Now, if your plasmid DNA is cut, that means your plasmid is wasted. So these single-stranded DNA would act as like baits. So instead of the plasmid DNA being cut, these will ensure that the nucleases cut the single-stranded DNA and in a way ensure your plasmid is prevented from cutting and it can enter the cell. And many people use this salmon sperm derived single stranded DNA for this particular process. So these are the possible mechanisms. So I hope this was clear. We'll go to the next question. Which of the following enzyme could be used to partially digest the cell wall of yeast cells? And four options are given. So here we'll talk about start with the wrong answers. If we can take a couple of minutes and then if anyone knows a particular answer is incorrect, you can justify why that answer is incorrect. We'll take a couple of minutes.
Sir,
Yeah, Vishwajit.
sir, option D is the wrong option.
We'll take a couple of minutes so that we'll start with you. You can start answering in 2 minutes so that we give others time to think. Okay? Yeah, Vishwajit, you can start.
Sir, Option D, lysozyme.
Okay. Why do you think it's wrong?
Sir, it is present in lysosome to degrade any waste, any organelle or
Let's say you know the
waste protein.
do you okay lysozyme, do you know the amino acid sequence of lysozyme? Do you know, as in, like, do we know, is it characterized?
No, sir, I don't know.
No, you don't know, but okay.
Uh,
Do you think people have worked on the structure of lysozyme? Can it be recombinantly produced?
no, sir, I have not read any article or anything
Okay,
about it.
okay, what you meant was that it is present in the lysosome. So people
Yes,
have
sir.
characterized it, meaning you know the amino acid sequence from which you can take the genetic sequence also. If that is the case, you can overexpress lysozyme and purify it and buy it as an enzyme. So usually
Yeah.
lysozyme is it comes as powder, lyophilized form. We can take it
Oh,
and we can make a solution out of it.
okay.
So lysozyme is available. It's not like it's not available. Let's say now it is available. Will it really help in digesting the cell wall of yeast cells? You're muted, Vishwajit.
Uh, no, sir, because it would degrade protein which is on cell wall. No, not cell wall. On plasma membrane of each cell.
Does the cell wall have proteins?
No, sir, no.
Okay. So that's how you need to think about this question. If the cell wall has proteins, then lysozyme will be helpful. But you say cell wall doesn't have proteins, so that means it cannot.
Yeah,
So that means it cannot really uh, help. Adding lysozyme will not be helpful. So then, what
yes.
does the cell wall have?
Um, Carbohydrates, peptidoglycan,
Huh. So,
and
huh. Sorry, I'll let you talk. Sorry. Yeah.
no, sir. Yeah,
Yeah. So the cell wall is usually made up of chitin. Chitin is N-acetyl glucosamine, and it's a polymer of N-acetyl glucosamine. You have this, then you have polysaccharides, then specific polysaccharides like mucopolysaccharides, and you also have other glucans. So this is what your cell wall is made up of. So because of which we can eliminate lysozyme. Does anyone else want to try and attempt answering? Any other option? Priya? Yeah, Rajita.
So option B, aldolase.
Aldolase. What does it do?
So it's an enzyme involved in glycolysis
Glycolysis. Yeah.
um, for catalyzing the cleavage of fructose 1,6 bisphosphate into glyceraldehyde 3 phosphate and next further steps like dihydroxyacetone phosphate.
Huh. So basically, it would break down your 1,6 bisphosphate into G3P and DHAP. Right. So unless your fructose 1,6 biphosphate makes up your cell wall, it can't really work. Right.
Yes, sir.
Right. What about other options? We have two more options left. Metalloproteases and zymolase. Yeah, Chandrama.
So option A, metalloprotease. Actually, these enzymes require metallic ions
Hmm.
to function. Its function is to lyse actually the peptide bonds using those metallic ions.
Lyse or hydrolyze?
Hydrolyze.
Hydrolyze.
It's
Yeah,
hydrolyze.
right. So because we don't have too much of proteins on the cell wall, we do have some, but it's not the proteins that is holding the cell wall. Rather, these are some embedded on the cell wall. So having any kind of protease like a lysozyme or a metalloprotease will not help. So you all say that zymolase is the right answer. So now, does anyone want to talk about how zymolase works? What is zymolase and how it works? Priya, do you want to try?
Zymolase partially digests the cell wall and makes spheroplast.
Hmm. What is it? Spiroplast, is it?
It's basically the yeast cell without the proper cell wall, I guess.
Okay. Okay. That is okay. I don't know what spiroplast is, but yeah, it digests the component of your cell wall. That is true. So here you can see that in this yeast cell called from the strain called Candida tropicalis, you have the cell wall here. That's the white colored membrane, followed by the cell membrane, which is thin here. And you have other regions like nucleus, mitochondria, vacuole. So after treatment with zymolase, you see that the cell membrane is visible, but the cell wall has completely been degraded. Now this will lies the glucans here, and it will break the linkage. So we were earlier talking about specific that like we were generally saying that glucans are present. Now this zymolase hydrolyzes a specific glucan called laminary pento hydrolase. Sorry, laminary pentose. Now this zymolase will break the linkage, so breaking down the cell wall. This is the specific activity of this zymolase. Ah, so the next question is, in the calcium phosphate method, the mammalian cells uptake the DNA calcium phosphate complexes by the process of, and four different options are given: endocytosis, pinocytosis, exocytosis, and macropinocytosis. Uh, does anyone want to explain or try to eliminate the wrong option? We'll start in a couple of minutes. We'll start by eliminating the wrong options. Does anyone know what you can Try to explain what any of these processes are. Yeah, Priya, do you want to go first? Yeah, Vishwajit. You can unmute Vishwajit.
Sir, exocytosis is process in which molecules are transferred from inside to outside the cell.
Right. Okay. Good. Rajita, do you know of any other options and what their that particular phenomenon is?
So B,
Pinocytosis.
pinocytosis,
Yes.
is the form. It's a form of endocytosis specifically for the uptake of extracellular fluid and dissolved substances.
Okay. Would that extracellular fluid include DNA?
Um, so these are liquids other than particulate matter.
Hmm. Uh, Now, did you mention that it is soluble, dissolved molecule? Did you mention something like that?
I mentioned extracellular fluid and dissolved substances.
Dissolved substances. So that means only if it is dissolved. And will your DNA plasmid be dissolved? As in, like, is it? Do you think it will be dissolved?
No,
Why do you think it will not be dissolved?
since it's a large particle.
Possible. That's how even I'm thinking about this. So, pinocytosis. Okay, how is pinocytosis different from endocytosis? Like, you can think about the way in which the particles are taken inside the cell. Do you think is there any difference between pinocytosis and endocytosis? Yeah, Vishwajit.
Sir, in endocytosis, usually it is receptors bind to the plasma membrane, and then it takes.
I can't clearly hear you, Vishwajit. Can you check the
Okay,
mic on your side?
now is clear, sir.
No, it's you. We still get that audio thing.
Sir,
Okay, it's okay. You can try again. Yeah.
sir, in endocytosis is uh, Clathrin mediated endocytosis and Kavlin mediated endocytosis.
Hmm.
Uh, those are receptor based endocytosis process are there.
Okay.
Yeah, I mean that only I know.
Okay. Do you know about vesicle formation?
Yeah, sir.
Right. So, yeah, you did mention about receptor-mediated endocytosis. Now, in both these cases, you have this vesicles that are being formed.
What, sir?
In both these cases, endocytosis and pinocytosis, you have vesicles that are being formed.
Yes.
But in pinocytosis, like it was mentioned by Rajata, the particle has to be either solubilized very well, like small solutes, but owing to the size of the DNA, it can't be called as pinocytosis because in pinocytosis it's majorly the liquid, whereas in endocytosis it is not majorly the liquid, it's the physical form. So that is why we don't call this method as pinocytosis or macropinocytosis, whereas we call it as endocytosis. So it's
Yeah.
was that clear, Rajata and others?
So pinocytosis vesicles are smaller in nature than endocytosis vesicles.
We can say that because usually in it in pinocytosis only the fluid and whatever is dissolved in the fluid is taken. So the kind of vesicle that is formed is not as large as other vesicles that happens in endocytosis.
Yes, sir.
And
Thank you.
one more thing is in endocytosis you have different ways where you can use receptors and other uh, how do I say, on the membrane you will have some embeddings that will help you to physically interact with substances that will basically fold inwards after something is stuck onto it. So with respect to DNA, that is the possible way in which it is happening. It is physically binding, after which the membrane is folded inwards. So because of this, we can say that this happens through the process of endocytosis and not pinocytosis. Was it clear or should I repeat this again?
So then what is the difference between A and C options?
Macropinocytosis might have been for larger solutes, but okay, how I understand this is your DNA physically interacts with your membrane. Once it interacts with your membrane is folded. If this is your membrane, if this is your DNA, once it physically interacts, it is folded inwards and the vesicle enters the cell. But with respect to pinocytosis, you don't have any physical interaction. There might be some DNA. If it is there in the extracellular fluid, it might be taken up as the vesicle is formed. But specifically in this process, you are looking at interactions of the DNA with the cell. Only if the DNA is touching the cell, it will form a vesicle, which is called as the endocytosis, and it is forming. Macropinocytosis is an extended version of pinocytosis where you are able to take up higher volumes. That's what I think is happening. So, why I think endocytosis is possibly the right answer is because of the physical interaction. If you think it's not right, you can let me know. We can go through this question at the end. Or if you think, or if you have any other reference, we can discuss that as well. We'll see. Did that answer your question, Rajita?
Yes, sir. Yes, sir.
Okay, fine. Then we can move on to the next question. Yeah, the method in which viruses are used for transformation in mammalian cells is option A, liposome method, option B, bacteriophage, option C, transduction, and option D, lipoplex method. Uh, if anyone knows about the right answer for this question, we can directly start with that.
Sir, it's option C, transduction.
Okay. Do you know the variations, Chandrima? What is transduction? What is transfection? What is transformation? Do you know the differences among these? Huh, you're right. It's transduction.
Actually, here the transduction, we are using the bacteriophage or any virus to insert the DNA.
Right,
If we are using transfection, then other than the virals, we are using to insert the DNA.
right. So, for example, uh, you'll have a DNA. You will coat it with something, but you will use mammalian cells, and you will ensure that whatever you're coating helps in your DNA to enter. Is that what you're trying to say for transfection?
Yes, sir.
Right. Yeah. So in labs we use this reagent called lipofectamine. So that basically what it does is it interacts with the lipids and helps in merging lipids or whatever is contained within the lipids. So earlier we were discussing about vesicles, you remember? Here, what happens is your DNA is engulfed within a vesicle-like stuff that's called the lipofectamine. Once this lipid surrounding or lipid, uh, or how do I say, once you have this DNA within this lipofectamine uh, thing, this lipofectamine takes it and it goes to the nearby lipids. And because your cell exterior has lipids, it can easily merge and fuse. Once this fusion happens, the DNA can enter. This is called transfection. I just wanted you guys to know about this, which is why I have this figure here and the differences. The next question is, in the bacterfection method, dash is the commonly used transformation of plant cells. Option A, Salmonella. Option B, Agrobacterium tumefaciens. Option C, Shigella. And option D, E. coli. We'll take a couple of minutes and then answer this question. Yeah, Ramesh, do you want to try answering this question? Yeah, Vishwajit.
Sir, option B is the right option
Okay.
because and it contains Ti plasmid,
Huh.
uh, which is generally used for transformation uh, in plant cells.
Okay. What is Agrobacterium tumefaciens? Do you know what it is?
Um, it is a bacteria.
Okay. Is it like host pathogen, something like that?
I mean, yeah, I mean, it wounds to a plant and then it transfers its plasmid.
So, was it it? What is it? Is it like an infection causing thing?
Uh, and, and I don't know about infection, but it wounds to plant and then I transfer its plasmid.
Okay, what is it gaining out of it? Why should something transfer, uh, how do I say? Why should Agrobacterium tumefaciens transfer its DNA?
I don't know, sir.
Okay, you can think about this as host pathogen interaction. So, okay, before that, does anyone want to answer this question? Priya, Rajata, Chandrima? Do you know anything about Agrobacterium tumefaciens?
So actually, it causes a tumor or the tumor cells in that plant.
Hmm.
They are having a DNA called tDNA, which they insert into the plant,
Hmm.
which cause that disease.
Okay. Okay. Fine. It's a. Can we call it a host pathogen relationship here between the plants and Agrobacterium tumefaciens?
Yes, sir.
Okay. Great. Okay. Now we've decided that Agrobacterium tumefaciens is the right one. We'll look at how it works. Like you mentioned, like you mentioned, it has a Ti plasmid in which there is a tDNA. Now we take our DNA of interest and put it in instead of tDNA. So now we have the vector which is made up of the Ti plasmid with our DNA, and we can introduce that to the cells for the plant cells to take up. So this is how it works. The next question is, in insertional inactivation mechanism, the successful cloning of the gene of interest in the LAC-Z gene will result in dash colonies. So in last week's lecture, we discussed about blue-white screening. Uh, so with respect to that, does anyone remember about the coloring and what happens? Does anyone know what LAC-Z will produce?
Beta galactosides.
Okay. What is that? What is beta galactosides?
It breaks down lactose into glucose and galactose.
Okay, it can break down sugars, specific sugars to give galactose. Now here, when we do these kind of experiments, we give it a substrate called XGAL, which in the presence of beta galactosidase will produce a blue precipitate. But here we introduce a new term called insertional inactivation. So what we mean by that is, instead of the LAC-Z gene, we'll place our gene. So that means if you give the XCal substrate, and if a colony produces blue precipitate, that means our gene of interest has not been placed inside. Rather, LAC-Z is active. So it is called negative selection. Was that clear? In these cases, instead
Yes,
of
sir.
getting blue colonies, we'll get colorless colonies. Okay. Huh. The next question is the CI repressor, when active, what does it do? It represses the lysogenic phase, it represses the lytic phase, plaques are observed, and or it represses the viral polymerase. Does anyone know about the CI repressor? Or we'll start with the different phases of viral growth. Can anyone list the both or the two phases of viral growth?
Lytic and lysogenic
Yeah.
phase.
So what is lytic phase?
It, I mean, uh, when transduction is been introduced in, I mean, sorry, DNA is introduced in bacterial cells,
Huh.
and then um, those bacterial cells is disrupted. I mean, those are live.
Okay. Why is it getting lysed? Do we know about it? Okay, I'll just talk through this. So you have a bacterial cell and your virus is entering. Once your virus is entering or trying to attack, it releases the DNA or it could be an RNA. It depends on what kind of virus it is. Once you have the genetic material, it will use the host machinery to both replicate the viral genetic material, could be DNA or RNA, and then also produce proteins. Now, when this production of proteins is happening, the bacterial cell is intact. There is no issue with it. After some time, it will produce enough proteins to make multiple copies of the virus itself. Now, this stage where it is producing these multiple copies of virus is called the lysogenic phase. Once it produces multiple copies of this virus, now these viruses can infect other bacterial cells. That is when the cell will break down, releasing multiple viruses. That is called the lytic phase. Is that clear?
Yes, sir.
Ah, okay. Now we'll come to the CI repressor. Does anyone know about CI repressor? And is there any other repressor that's involved in this particular system? Okay, fine. I'll just talk you guys through. So what you see here is based on the crystal structure of this lambda repressor, also called as the CI repressor, because it comes from the CI gene. So what you see here is a DNA, and you have this dimer of lambda repressor with the NTD regions, which is N-terminal or amino terminal domain binding to the DNA. So you have this dimerized lambda repressor that binds to a particular operator. When it binds to this particular operator, this RNA polymerase cannot move beyond this. So the CRO genes and other genes will not be expressed. This happens during lysogeny. During lytic phase, this CRO proteins, which can bind to the other operator region, will prevent lysogeny. And only those genes that need to be expressed for the lytic growth will be allowed for expression. So that is how the switch between lytic and lysogenic phase happens. Uh, so answering this question, the CA repressor, when active, when CA repressor is active, it will bind to the operator before the CRO region. So that time it will repress this CRO region and it will repress the lytic growth or the lytic phase. That's why it's option B. Was it clear?
Yes, sir.
Okay. Yeah. The next question is the cytotoxic compound added to the selection media for URA3 mediated negative selection. So before going to this URA3 mediated before going to cytotoxic compound, does anyone know what URA3 mediated negative selection is, or what is URA3 selection? Chandrima, Rajata, Priya.
Are these media used in selection of yeast cells?
Yeah. So what do you know? Yeah, it is used for the selection of yeast cells. Now, do you think it codes for a protein or something? Can you build up on that?
I know it is essential for the biosynthesis of uridine.
Uridine. Okay. Hmm.
So it's involved in pyrimidine biosynthetic pathway.
Okay. So can it be? So when we talk about selection, we talk about positive selection and negative selection. So does URA3 can it be used for both positive and negative selection? You're right about the biosynthesis path. So this will code for this enzyme called ODCase that will help in pyrimidine production. So if this ODCase is not present, if it is a negative cell, then cell cannot grow unless you provide uracil in the media or uridine in the media. But if URA3 is present, then the cell can grow in the absence of uridine. Now, what they say is when this 5-FOA, 5-fluoroauric acid, is added to media, which is an analog, this ODCase will convert this 5-fluoroauric acid into a toxic compound called 5-fluorouracil that will basically bind to your RNA polymerase and will reduce the transcripts. So this way, it can allow for selection against yeast that carry this gene. So now with respect to the question, they ask that which cytotoxic compound is added to the selection media for URA3 mediated negative selection. So 5-fluoroauric acid is that compound. So was this clear or okay, I'll rephrase it. Now what you need to know is uh, you have this URA3 gene. Just imagine it in a setup similar to LAC-Z. Instead of LAC-Z, imagine this as URA3. Now you want to put your gene of interest into this. In some cases, your gene of interest will be replacing lac Z. And in those cases, if you give uridine, those cells that can take up uridine can survive. In other cases, you want to see if your URA3 gene has been replaced. So, if it is not being replaced, then the URA3 gene will code for this ODCase. And this ODCase can convert this 5-fluorouric acid to produce 5-fluorouracil. And that way, you can confirm whether your gene of interest can replace has replaced this ODCase producing gene called URA3. Was that clear? If not, I'll try repeating again.
So can you repeat again?
Yeah. So what you want to do here is you want to see you have whether the gene that you have whether it is replacing your URA3 gene or not. How can you do this? When you know that URA3 gene is essential for uridine to produce. Now, how we saw in LAC-Z about the negative selection to get colorless colonies, similar will be a method here. What we will do is we will give 5-fluorouric acid. When we give this, if there is this ODCase, that is a URA3 gene, if that is present, then 5-fluorouric acid will be converted to 5-fluorouracil and the cell will die. So only those that do not have this or those that have it supplemented with some other, only those can survive. Others will die. This way, we will eliminate those cells that have this URA3 gene intact within the uh, selection plate. So that is the logic here. I have also attached link to a particular paper. If anyone is interested, you can check the paper for how this URA3 gene acts. The next question is, which of the following reaction is catalyzed by the gene product of 4A gene? Uh, Now 4A gene, if you look on UniProt, I've attached the link here. It codes for alkaline phosphatase. So what it does is, it can basically remove a phosphate from a phosphate monoester or a phosphate diester. It could be anything. Uh, how is this relevant to the topics that we have been discussing? In cloning, what we do is we generate a double digested plasmid, then we have our DNA of interest. Now, this double digested plasmid, if you put the ligase enzyme, there are possibilities based on the sticky ends that this double digested plasmid will form, will be ligated, self ligated to form a closed circular DNA, which is a false positive. So to reduce this chances of false positive, what we do is we treat this double digested plasmid with phosphatase. So the 5' phosphate will give us a 5' OH. Now, only the DNA insert, which has the compatible sticky ends and the overhangs, will sit, and the ligase will work. So this is the relevance of the alkaline phosphatase with respect to cloning, whatever we were learning earlier. Was this question clear?
Yes, sir.
Okay. The next question is di-deoxynucleotides are used in, and they've given 4 different methods: Maxam-Gilbert method, Illumina sequencing, Sanger sequencing, and next generation sequencing. Does anyone want to try answering for this question? Where is di-deoxynucleotides used?
Sir, option A.
Maxim Gilbert sequencing. Okay. What happens there, Vishwajit?
I don't know entire procedure, but um, um, but on when we on the electrophoresis, um, sir, I I don't. I mean, I do not remember.
Okay. So, dideoxynucleotides is not used in Maxim Gilbert method. In Maxim Gilbert method, you make physical modifications, but in Sanger sequencing, that's where you use this dideoxynucleotide to reduce the length of your DNA product with which you identify that particular base. So, what specifically happens is it's shown here. I'll give me a moment. Sorry. So, you have a template DNA and you have a primer, and you will put it with different DNTPs and a particular double, dideoxy NTP. Now, you know that for the polymerase to extend, you need only DNTPs and not dideoxy, because if you have dideoxy, your phosphodiester bond cannot form. Is it clear till this point?
Yes, sir.
Okay, so what you see here is the physical difference. This is your deoxy ATP, and this is your dideoxy ATP. In your dideoxy ATP, you don't have a OH. If you don't have a OH, your phosphodiester bond cannot form. Now, when you put both of, so in your reaction, what you will do is you will choose three of these, and for the fourth one, you will use a, uh dideoxy NTP and some amount of the deoxy NTP also. So, what will happen is at that particular location, this dideoxy NTP will bind and it will prevent, or it will terminate the chain's propagation. So at particular length you will have these. Based on this, you will run it through a capillary and you will hit it with a laser. Now these are labeled. Based on the particular labeling and the length, we'll be able to identify the entire sequence. And this is how Sanger sequencing is done. Was this clear?
Yes, sir.
If not, I can repeat.
Sir,
Should I repeat? Yeah. Yeah, Vishwajit.
uh, the laser detection, is it through flow cytometry?
Okay, I'm not sure if it's through flow cytometry. I haven't done this, so I'm not sure. But what I know is you have a every dideoxynucleotide has a particular fluorophore that it is attached with. So you have specific detection, but I'm not sure if it's through flow cytometry.
Okay, sir.
Right. And the last question is, in the G reaction of Sanger sequencing, the DNTPs are, and four options are given. So anyone who knows this can try answering this question. In the previous slide, we discussed what exactly will be the changes.
Sir, option B.
Yeah. So this is the one that has dideoxy-GTP. So we'll use this. Yeah. So with this, we come to the end of this week's problem-solving session. If there are any doubts, we can discuss, or we can end the class here today. You can unmute if you have any queries.
Sir, it is not about on the the course.
Huh.
I have a general doubt.
Yeah.
Sir, do you know something? I mean, like on how to crack gate exam?
I'll just finish this recording. We'll discuss that after the recording is over.
Okay, sir.
Yeah. Others, do you have any questions? If not, I'll just end the recording. We'll stay on the call. Okay, I'll end the recording.